So we're transitioning today to a new system, to the renal system, from the respiratory system to the renal system, the urinary system. Obviously, we've already talked about kidney a little bit in the respiratory system, and one of the goals is to then make this integration of knowledge at the cellular and molecular level of the kidney and bring that into alignment with your knowledge about the cellular and molecular mechanisms in the lungs. So, new section, so we're going to start with anatomy, both at the gross and the macroscopic level, and then we'll get to some microscopic level anatomy. Let me remind you there's a quiz this weekend. The quiz will cover the material from class today and Thursday. It will be available after class on Thursday. It will mostly anatomy, but not exclusively anatomy. It's the material we talk about in class today and Thursday. Same deal, pick it up, take it, close book, put your electronics away, turn it back in at the CST desk, maybe a half an hour. What did I say about it? Oh, it'll be due on Sunday. Rather than Monday, let's make this one due on Sunday because that gives me time to grade it and then hand it back to you on Tuesday. So, kidney. Let's just start talking about use some of the anatomical language related to the kidney. The first thing to realize is that the kidneys they're called, it's said, the word is retroperitoneal. They are behind the peritoneum. And they are much higher in your body than you think. You think of the kidneys as being, sort of down here in your pelvis, at least I do. Oops. Do we want to get some paper towel? They're up under the ribs, so they're almost this high. You're going to see this in the lab this week when we start digging into the abdomen of the cadaver to look for the kidneys. And let me also be clear that from an anatomy perspective, it would make sense to do the digestive tract before we do the kidneys because the digestive tract is anterior and the kidneys are posterior. But from a physiological perspective, it makes way more sense to do the kidneys now from the respiratory system and then go to the digestive tract. So physiology and anatomy here are not aligned well with regard to the cadaver. So we're going to push the digestive tract aside physically to get to see the kidneys in the cadaver. And we'll also have pig kidneys to look at, fresh, frozen kidneys. No, pig kidneys. Okay, so let's just start using some of the language of anatomy. Here's the kidney, right? Kidney shape. It receives a robust blood supply from the renal artery, which is a branch off of the abdominal aorta. We'll see that when we open up the abdomen more. The renal artery feeds a lot of blood to the kidneys. I'll talk about blood flow in a minute. And then the renal vein drains the kidneys up from the blood that has been processed and goes into the inferior vena cava. So it's right off the abdominal aorta, kidneys, processing, and then right to the inferior vena cava. The urine that is collected comes out of a tube called the ureter. The ureter goes to the urinary bladder. Well, we're not going to talk too much about the bladder. We'll probably look for it in the cadaver, but we're not. The bladder is just a muscular bag, not that interesting. So the ureter is the tube that brings urine from the kidneys to the urinary bladder. The kidney has kind of three layers. The outer layer is called the cortex. So, you know, it's not a brain cortex, this is a kidney cortex. The middle layer is called the medulla. It's not the brain medulla, it's the kidney medulla. Cortex just, I think, is Latin or maybe Greek for rind, like the rind of an orange. And medulla is Latin, I think, for middle. Cortex, medulla, and then in the center is an area called the pelvis, but it's not the hip, it's the renal pelvis. And the renal pelvis is what's going to collect the urine before it goes into the ureter. So the pelvis is physically connected to the ureter. Okay, so here's some, but not all of the words. Let's talk, well, actually, let's do the anatomy first, and then get back to physiology. No, I'll just follow the order, sorry. So let's talk about physiology for a minute. What does the kidney do? So the kidney is essential for conservation of water, regulation of body water. If you become too dehydrated, you don't survive. If you're too hydrated, actually, it's not ideal. People have died from overhydration. So the kidneys are trying to maintain a correct level of hydration by either retaining water or excreting water. So water is important. Inorganic ions. So what do they mean by that? They mean sodium and potassium and calcium. Maintaining the body's concentration of the critical ions. Phosphate, chloride, a little less important, but also critical. But all the ions that we've been talking about, sodium, potassium, calcium, chloride, the kidney's job is to maintain homeostasis of those ions. And then we've already talked about acid-base balance, right? Keeping the pH where it belongs by either doing metabolic compensation, doing renal compensation for either respiratory acidosis or respiratory alkalosis. So, and you can imagine we're going to see a Davenport diagram again. This one, removal of waste products, is the one that you naturally think about. You know, the urine is a disposal mechanism for waste products, and we'll talk about their removal and excretion. And we won't talk too much about foreign chemicals, you know, xenobiotics, drugs, other things that aren't nutrients, are not metabolic, but still need to be removed from the body. We'll talk about that some on Thursday. So the overview of the kidney function. Biggest possible picture from the physiological perspective is we're going to take that blood, a large amount of blood, so approximately 1/5 of the cardiac output goes to the kidneys. So you may remember that cardiac output is about 5 liters per minute. So about 1 liter per minute of blood is going to the 2 kidneys. So it gets a lot of blood. We're going to filter water and small molecules from that blood. And we're going to filter a lot of it. 100 mils per minute. So just imagine, you know, let's imagine, you've all seen what 100 mils looks like. I mean, you've held a 100 ml beaker in your hand or a 100 ml graduated cylinder in your hand. 100 mLs, 100 milliliters is not a small volume. And that's every single minute. So that's what's getting filtered from your blood. A large volume of plasma, essentially. Then we're going to, the kidneys, our kidneys are going to recover. Reabsorb is the official word. Recover or reclaim most of what we want. We want to conserve water. We want to recover water. we want to recover ions, we want to recover glucose that's in the blood. So we filter it out, and then we recover what we want. And then the rest gets excreted. It doesn't seem like the most rational mechanism for it, but it's what we have evolved. It allows us to get rid of everything and then recover what we want, what we need. It works. Obviously it works. Let's talk for a minute about this volume. So 100 mils per minute. So when you void urine, when you go to the bathroom, you probably urinate a couple of hundred mils, right? Maybe 200, 250, depends on how long it's been since you last went to the bathroom. So here we're saying that the urinary fluid, the fluid that is getting filtered, is approximately the same amount that you urinate every single minute. So that's a large volume. If we didn't recover 99% of it, you'd be spending every minute in the bathroom, every 2 minutes, every 3 minutes in the bathroom. And you'd be losing you'd be drinking and urinating all the time, basically. So obviously that's not going to work. The point is, this is a large volume, and so we need to recover almost all of it so that we can excrete a small volume of urine, about 1 mil per minute, which means you can go 2 hours, and that's 120 mils of urine. You can go 3 hours, and that's 180 mils of urine, plus or minus. Depends on whether you've been drinking a lot of water or coffee or not. Okay, key points. Filter it out, recover what we want to retain, what we need to retain, and excrete the rest. And we're going to be seeing the molecular mechanisms of those processes over and over and over again. So now let's go back to anatomy. The kidneys are retroperitoneal. They're relatively high in the abdomen. They receive blood from the abdominal aorta, and their venous blood drains into the inferior vena cava. That's the main point of this slide. We have 2 kidneys, one on the left and one on the right. They're both essentially the same. The one on the left is a little bit more superior than the one on the right, but that's not big. So let's now talk some more about anatomy. So this is going back to that first slide. Cortex. Medulla is the layer. There are 2 structures in the medulla. So there's really one structure in the cortex. You can see it's pretty homogeneous. There are 2 structures in the medulla. And then then the pelvis connected to the ureter, which connects to the urinary bladder. The functional unit of the kidney is called the nephron. So just like the neuron is the functional unit of the nervous system, the nephron is the functional unit of the kidney, but it's not one cell. It's thousands of cells. So the nephron, made up of thousands of different types of cells, is the functional unit. Okay? Now, so here's the slide I think I printed out for you that covers most if not all of the anatomical terms that you are responsible for knowing for the quiz and for the exam. Okay? So let's go through them. And I'm going to go from outside to inside because that's what I've been doing. So the layer on the outside, the outermost sort of surface, is called the capsule. It's just a name for the surface membrane on the outside of the kidney. Then the next layer is the cortex. So this whole layer here is called the cortex. Okay? Then this whole layer is called the medulla. And it's comprised of 2 structures. and only one of them is labeled on this slide. So you're going to decorate your slide with one additional label. The medulla has structures that are called pyramids. This structure is called a pyramid, and you'll see it in the lab on Thursday. And the areas of the medulla between the pyramids are called columns. So the word that is missing from this slide that you need to illustrate yours is that this area right here, and this area right here, and this area right here are renal columns. So the medulla has pyramids and columns. The inner tip of a pyramid is called a papilla. It's just Latin for nipple. And you'll see it's a little nipple structure on the innermost section, innermost part of one of the renal pyramids. Okay, so pyramid, column, pyramid, column, pyramid, papilla, pyramid, papilla. The papilla is the junction between a pyramid and what are called minor calyces. So one calyx, this is Greek, one calyx, two calyces. This is not a Greek class, so if you don't use the plural right, I'm not sure, but just to be clear, one calyx, two calyces. So there are minor calyces that are the contact point with the papilla of the pyramid. Then the minor pyramids are connected, the minor calyces, one minor calyx is connected to a larger structure called a major calyx. The major calyces are connected to the pyramid, to the pelvis. Okay? The blood flow, the renal artery is going to provide blood to the cortex and the medulla, and we'll talk about the microscopic circulatory system in a minute. Renal artery feeds blood to these structures. The renal vein is going to drain blood from these structures, and both the renal artery and the renal vein enter the kidney in a structure in the sort of the concave, the most concave part of the kidney called the renal sinus. Any questions about these terms? Like with any anatomy, just using it, maybe my recommendation is draw a picture of a kidney and then start labeling it, from memory. Well, first do it by just sort of tracing it, and then do it from memory. Use the coloring book. The coloring book assignments are helpful. Yes?
How big is like a kidney actually?
That. You will see it in that. I think this picture is pretty correct, right? Same dimension as the heart in the up-down position, maybe this thick, this tall. Okay, so you know what this means. This means we have a game. So I'm going to So this is our anatomy game. You know how to play. Who wants to start? So what you do is you choose a name, point to a dot, and then we'll see if you get the happy ding. Okay, you're in. What are you calling that?
Oh, renal cortex.
Renal cortex. You want to go to this one?
Yeah, oh wait. Yeah. Actually, no, that's the
Okay, so do you want
capsule. Can I go to renal
capsule to this one? Capsules, and so what it's pointing to is the outermost layer.
This one, renal artery?
Yeah, so the color obviously helps. Well, so that is, there is no dot for the renal artery, but that's the renal artery and renal vein. If on a quiz, I ask you to identify the renal artery and the renal vein, and it is in black and white, obviously I'm not going to ask you to know which is which, because you can't tell which is which from a simple diagram. Although, if I gave you a physical specimen, you should be able to tell, because remember, arteries are thick-walled and veins are thin-walled. So you could tell the difference. If I gave you a hint on a quiz, if I said, this tube has a thick wall and this tube has a thin wall, then you could say which was the artery and which was the vein. So this dot, you're saying this dot is the pelvis. Okay.
This one should be
Okay. So
if I'm seeing
notice-
it correctly.
notice that this little,
Yeah.
bar thing, I don't know, TIE fighter, whatever it is trying to point to the layer. So that would make that the cortex.
The renal pyramid.
Renal, so it's pointing to the layer. It's using this bar thing here as pointing
As
to
the
the name of
whole
the layer, the
Okay,
whole
so
layer.
then the medulla.
Right. Yeah, obviously on a quiz, I would accept both pyramid and cortex for that one, because it is pointing to a pyramid as well.
The renal column.
Right. Renal column is the space between pyramids. Not space, it's material. Yeah. So this thing, so this line is pointing to this structure here, which is the renal pyramid.
Would that be the major calyx?
So this, major. You're saying this would be the major calyx,
Yeah.
and so the thing connecting to the major calyx, the multiple things that are connecting to a major calyx are minor calyces. Okay? So this one wasn't so hard, but this is another tool you can use to reinforce your anatomical language. Yeah.
So for the major calyx, is there a plural for a major, or is it all just one?
No, there's multiple. So here's a major calyx, and here's a major calyx, and here's a major calyx. So we have multiple major calyces, and each major calyx is connected to multiple minor calyces. There is one pelvis per kidney. Okay, so you now have this resource to study from, and as well as the coloring book and the other ways that you learn anatomy. Okay, so now let's transition to the microscopic anatomy a little bit before we start talking about the function, the physiology. So let's get oriented here. Here's the medulla. So this is the middle layer, and here's the cortex out there. So the capsule would be out here somewhere. Capsule, cortex, medulla. And so a papilla would be down here somewhere. Okay? The junction between the cortex and the medulla is where there are branches from the renal artery and the renal vein. This is not the renal artery anymore. It's a branch of the renal artery. And then it branches some more. So remember that this is a branch of the renal artery. It's not the renal artery itself. This branch, the renal artery has gone to multiple places in the kidney, and when we start to look at a nephron at this level, we're looking at a branch of the renal artery. Okay? So I've looked at these pictures a lot. Here is a nephron. The yellow and some of the red structures are really the nephron. That is one nephron. And here is another nephron. Okay? So this image is showing two nephrons. And it is showing two of the different types of nephrons. This is a cortical nephron because it's mostly in the cortex. So this is a cortical nephron, and this one is called a juxtamedullary nephron. It's a nephron that is at the junction between the cortex and the medulla. Juxta means near. Juxtamedullary. So there are two types of nephrons: cortical nephrons and juxtamedullary nephrons. They are mostly the same, but we will talk about one major difference between them later on, next week actually. From an anatomical perspective, you can see that this loop that it makes, that the nephron makes, which has a name, is called the loop of Henle. When I printed this, I tried to print it big so that you could actually read. Can you read? I didn't look at the copies. Can you read the word Henle on your copy? Okay, good. Because when I printed it out too small, you can't read anything. So this loop has a name, and it's called, which you will be responsible for knowing. It's part of the microscopic anatomy of the kidney. This is the loop of Henle. The juxtamedullary nephrons, the loops of Henle, are long, and they go deep into the medulla. The cortical nephrons have short loops of Henle, and they may not go into the medulla at all, or if they do, they penetrate very shallow into the medulla. That's the main difference, and there will be a functional difference that we'll talk about next week. So let's just look at the cortical nephron here. And let's follow blood and urine. Blood is what's fed in, urine is what comes out. I'm going to use anatomy to try to tell the story of processing of blood to make urine. So blood enters this nephron from a branch of the renal artery. And then the blood enters a structure called the glomerulus. That's got to be on here somewhere. Okay. So you have to annotate your slide with another word.
It's on the left.
Is it at the top?
It's like from the top.
Oh yeah, there it is. Okay, so it's labeling the juxtamedullary nephron. So this is one of the most important structures of the kidney, because this is where filtration happens. This is where that filtrate of blood comes out of the blood and goes into what's going to become urine. So there are arterioles here, right? So blood is going to enter the glomerulus in the afferent arteriole. The afferent arteriole is what brings blood in. Afferent arteriole is what brings blood in. Blood is going to enter a network of glomerular capillaries. So in the glomerulus, there's going to be a network of capillaries called glomerular capillaries. And then arterial blood is actually going to leave the glomerulus, the glomerular capillaries, in the efferent arteriole. Afferent, now it's just, afferent means going in, efferent means coming out. You know what an arteriole is. Afferent, blood goes in, efferent, blood comes out. So this is an interesting capillary bed. This is a capillary bed, but what comes out is not a vein. What comes out is another arteriole. Then the blood comes out of the afferent arteriole and then enters another network of capillaries called the peritubular capillaries. So this blood is processed through two capillary beds. The glomerular capillaries, where filtration happens, and the peritubular capillaries that then are part of the processing system, and then the peritubular capillaries give rise to venules, which go into little veins, which go into a branch of the renal vein. So it's a more typical capillary bed. Arterioles going in, capillaries, veins coming out. Okay, so that's the story from the blood perspective, using the anatomy. Bless you. Filtration is happening here. The filtrate, the fluid that was filtered out of blood, is collected into a space called Bowman's. So Bowman's capsule. So just like the name of the word on the outside of the kidney, the thing that surrounds a glomerulus is called Bowman's capsule. Bowman's capsule surrounds the glomerular capillaries. And so what comes out of the glomerular capillaries is collected into a space between the glomerular capillaries and Bowman's capsule, and that space is called Bowman's space. So Bowman's space is the space between the capillaries and Bowman's capsule. That's where the filtrate first happens. Remember, the filtrate is just a fluid that comes out of blood. At this point, it's very similar to blood plasma. And we'll talk about the processes of filtration in just a minute. But it's a clear fluid, no cells typically, just plasma fluid. But we don't call it plasma anymore because it's not in the blood, it's now a filtrate. So the filtrate is collected into this Bowman space. Bowman space is connected to a tubule. And the first tubule is called the proximal tubule. The proximal tubule. Proximal means near. What is it near? It's near the glomerulus. The proximal tubule is the first tube. It's an epithelial tube that is going to process the filtrate. And so now we don't call it a filtrate anymore because it's being processed by the tube. And we call it urinary fluid. It's not urine yet because it hasn't finished being processed. It's no longer filtrate, so we call it urinary fluid. So, blood came in, afferent arteriole. Entered glomerular capillaries, got filtered. Fluid collected in Bowman space, and then starts to flow through the proximal tubule. Your textbook calls it the proximal convoluted tubule. that's fine. It's the proximal tubule or the proximal convoluted tubule. Both of those names are common. You'll see for a minute why they add the word convoluted in just a minute, but not yet. So then the proximal tubule processes the urinary fluid and starts to recover the things that need to be recovered. The water, the glucose, the salts. It started, the reclamation process, the recovery process, starts in the proximal tubule. Right away. Then the proximal tubule gives rise to the loop of Henle. And there's two parts of the loop of Henle. There's the descending limb, the part that's going deep into the medulla, or it's just the descending limb here is, well, it's hard to follow the convolutions. Let's just say this is the descending limb. And so descending limb and ascending limb. Now there are some distinctions. There's a thick segment and a thin segment. And I am not going to ask you to differentiate them. But it's obvious on a cartoon, they draw it thin and thick. And they do the physiology is slightly different. And next week when we do some histology, we will see some cellular differences between a thick ascending segment and a thin ascending segment. But I don't want to, your cerebral cortex shouldn't spend a lot of energy, don't spend a lot of cognitive load discriminating between thick and thin, okay? Let's just not do that. You do need to know loop of Henle, and you need to know, okay, well there's a descending and an ascending part, which is called limb, okay? This structure is going to be critical for the formation of a concentrated urine. So when we are dehydrated and our urine we need to recover as much water as possible, this is the structure we're going to be talking about. So filtration happens at the glomerulus. Reclamation, recovery, happens in the proximal tubule. Water recovery, specifically water recovery, is going to happen in the loop of Henle, which then goes into a distal tubule. So this is a distal tubule, and again, your text calls it the distal convoluted tubule. There's a part of it that is called the distal straight tubule. Again, don't spend any cognitive energy focusing on convoluted versus straight. If you see the word if you see distal straight and distal convoluted, it's all distal. That's the key point. Distal. Which is a little bit of a misnomer. It's close to the glomerulus. It's physically close to the glomerulus, but the fluid that went through it is now far away from the glomerulus, and that's why it's called the distal tubule. The distal tubule connects to a collecting tubule. Your book calls it a duct. I prefer the word tubule because tubule suggests that processing happens, which reminds me to say the distal tubule does some more processing of the fluid, as does the collecting tubule. And there's a cortical collecting tubule and a medullary collecting tubule. And by the time, so what's this connected to? This is connected to the papilla, which is connected to a minor calyx, which is connected to a major calyx, which is connected to the pelvis, which is connected to the ureter. So by the time we get out of here, we're talking about urine. This is urine. Let's see, so there are names here that your book uses that I would like to ask you, you can cross them out. Not that they're not important, and they are right, but the glomerulus and the Bowman's capsule are sometimes called the corpuscle. I don't use that language very much, but just so you know, glomerulus plus capsule equals corpuscle. Corpuscle, there it is, capsule and glomerulus, under the corpuscle. Just clarifying what your book is using. This whole thing, capsule and glomerulus, is the corpuscle. The loop of Henle, at the junction between the ascending limb of the loop of Henle and the distal tubule has a special structure called the macula densa. This point, the junction between the ascending limb of the loop of Henle and the distal tubule is called the macula densa, and it's an important contact with the glomerulus again. So you can see there's a contact here between at the macula densa. And that's going to be an important sort of feedback control point. So I think I've covered all the words here in this cartoon of two nephrons. And I've tried to give you the language of the anatomy and introduce some of the principles of physiology at the same time. Question?
Is it only in pyramid or also in columns?
These nephrons, the medullary nephrons, exist in the columns as well. These nephrons only exist in the pyramids. Well, actually, I should say, the pyramid, the medulla, this structure here, the loops of Henle, dominate in the medullary pyramid. The cortex, including the columns, is mostly glomeruli, or let's just use the words right. They're mostly corpuscles, proximal, and distal tubule. Now did that answer your question? So in the columns are really just from a functional perspective, a column is just like cortex. It's really an extension of the cortex. From an anatomical perspective, it's part of the cortex that has sort of penetrated into the medulla. So that's why, for example, in slides like this, no, this one's not easy to see, but in slides like this, the column is connected to the cortex. Other question? Oh, there's one thing I haven't mentioned. So let's go back to blood flow. Arterial blood, afferent arterial, glomerular capillaries. Let me go over here. Afferent arterioles, glomerular capillary, efferent arterioles, and then the efferent arteriole gives rise to either the peritubular capillaries, around, peritubular capillaries, or this loop of capillary that has a special name called the vasa recta. Vasa recta. The vasa recta is a loop of blood that surrounds and parallels the loops of Henle. So this slide, or this figure, makes it look like the vasa rectas are separated from the loops of Henle. But in 3 dimensions and in real life, these things are merged together. So the vasa recta is going to be important for reclamation of fluid, water, from the loop of Henle. Just like the peritubular capillaries are going to be important for reclaiming the good stuff that we recover from the proximal and the distal tubule. Remember, it has to get back into our blood so that it can go to the rest of our body. We don't want to just have it sit there in the kidneys. The water, the salts, the glucose gets recovered in the kidneys, and then the bloodstream has to distribute that to our brains and to our muscles and the rest of our body.
And does the recovery happen only in cortex? Like recovery of water and so?
No. The recovery of water, the recovery of most salt and almost all of the glucose happens in the cortex. The recovery of a lot of the water happens in the medulla. The efferent arteriole? The efferent arteriole doesn't really recover water. The efferent arteriole is what comes out of a glomerular capillary. So the glomerular capillary, I don't know how else to say it. So the efferent arteriole is what comes out of the glomerular capillaries and then gives rise to, is connected to peritubular capillaries or vasa recta.
Oh, yeah. What does
The peritubular capillaries are going to recover the water, salts, and glucose that the proximal tubule and distal tubule gives them by cellular and molecular mechanisms, and you can imagine it's going to get messy, and that's your future. And the vasa recta recover the water that the loops of Henle give them. I'm just giving you a heads up. The kidney's more complicated than the lungs.
Does the processing of the urine or the urinary fluid, does it stop when it's out of the collecting tubule?
Yes. By the time it's in the medullary collecting tubule, it's done being processed and it's now urine. There is still some processing in the cortical collecting tubule, but by the time it's deep into the medulla, we're talking urine.
Can you say cortical nephrine major role is recovery of the salt, glucose, and the juxtamedullary nephron major role is recovery of water?
That's not exactly right. The cortical nephron is important for the recovery of salt, water, glucose. Yes. The juxtamedullary nephron is important. Its functional role is the recovery of salt, water, and glucose, with an special part with the recovery of water, especially in a dehydration situation. So what you said was that this was focused on water, and it's not. It has a special adaptation, special for water recovery. But it does, this part of this nephron does everything that this part of this nephron does. So here's a slide which I printed for you, but I don't think you can read it, it's so small. So this is just a sort of an order of operation, and then we'll take a break. The glomerulus filters fluid into Bowman's space. So I'm just using the language again, but now we're not seeing the anatomy, so it's focusing on the physiology, and it's just a little simpler. The glomerulus filters fluid into Bowman's space. The fluid, the filtrate at this point, is processed by the proximal tubule. And here's a slide. Do not focus on the difference between convoluted and straight. Proximal tubule. Then the descending and the ascending limb of the loop of Henle. Again, don't focus on thick or thin. Proximal tubule, loop of Henle, processes the urinary fluid. It's not filtrate anymore, but it's not urine yet either. Then the distal tubule processes that fluid some more. The collecting tubule processes that fluid some more. And then by the time we get to a papilla, really, we're talking urine. Okay, so this is the order of operation for the urine. You should be able to do the same thing. What's the order of blood flow? I don't have a slide, so let's see if we can do this together. Okay, so afferent arterial. What's next? Glomerular capillary. Okay, what's next? Efferent arterial. Okay, so now you can see what I'm going to do here. What's next? Peritubular capillaries. And what would go over here? Vasorectal. The blood goes either one way or the other, and it depends on the nephron, and it depends on the branches of the capillary, that come off of the efferent arteriole. Okay, now I'm going to get blue for veins. Oh, what is upstream of the afferent arteriole? It started too late. Or let's just call it an artery. And what would go upstream of the artery would be the renal artery. So this artery is a branch of the renal artery, which then gives rise to the afferent arteriole. I just want to point out that a lot of students label this artery as the renal artery, and I take off half a point for that. Okay, so then the peritubular capillaries give rise to venules, and which give rise to a vein, which produces what? Not produces, what's connected to a vein that's coming out of the system. Like the renal vein, which is connected to inferior vena cava. And the same thing happens here with the vasorectal. Gives rise to veins, venules, vein, a vein, renal vein, inferior. Okay, so that's the order of operation of blood flow. The glomerular capillary is where the urinary flow and the blood flow connect, because this is where the filtrate happens. Okay, let's take a break and we'll talk some more about filtration, the molecular mechanisms of filtration. As always, the answer key will be, but is not yet, posted on Moodle, so in the next couple of days you'll be able to look at, I think you can see where you lost points, but you'll see the answer key on Moodle. And as always, I'm happy to answer questions about what did you miss. Okay, filtration. Talk about the molecular mechanisms of filtration. First, from an anatomical perspective. So remember, we're in the glomerulus. We're in the corpuscle, if you want. So here is the Bowman's capsule. Here are the glomerular capillaries. And here is Bowman's space. So let's just follow blood for a moment. Afferent arterial blood enters from an artery into the afferent arteriole and enters the glomerular capillaries and exits out the efferent arteriole. This is the contact between the distal tubule and the glomerulus, and you can see there are cells of the macula densa and some other cells called juxtaglomerular cells that form this contact between the distal tubule and the afferent arteriole. So we'll talk about that feedback contact site later. Afferent arteriole, glomerular capillaries, efferent arteriole. Filtration happens in the glomerulus. The cells that surround the capillaries of the glomerular capillaries. So remember, capillaries have endothelial cells, but the cells of the glomerulus have a specialization cells that surround them are called podocytes. So that's a cell name that I do want you to know. Podocytes are cells that wrap the glomerular capillaries. The blow up is, the zoom in is here. Here is a podocyte wrapping, so there's the cell body of the podocyte. Here is a process of the podocyte. It's wrapping the endothelial cell of the glomerular capillaries. And the point is, this capillary and these podocytes are leaky. This is not a tight tube. This is a leaky tube. And it's leaky so that water can be filtered and small molecules can be filtered. And there are structures of the podocyte wrapping around the endothelial cell of the capillary that are important for this leakiness. There are structures called fenestrae. Those of you who know Latin know that this is Latin for window. So there are fenestrae on the inside of the capillaries, and there are filtration slits. The podocyte has sort of fingers, these finger-like connections between one podocyte and the next one aren't tight. They aren't even physically in contact. There are slits between the one podocyte and the next that make it leak. So the fenestrae and the filtration slits is what's allowing fluid to pass from the capillary out into Bowman space. What forces fluid to flow? What is the force that forces fluid to flow from capillary into Bowman space? Good answer would be two words. First word begins with a B and the second word begins with a P. Blood pressure. Blood pressure is what forces fluid to flow through the leakiness. So let's just look at some actual, the cartoon is a pretty good representation of the way it actually looks, but let's just look at some real micrographs. So here is an AF. What do you suppose AF stands for? Afferent arterial. GL. What do you suppose GL stands for? Glomerulus. Efferent arterial. Here, I don't even know what ILU stands for, so I really don't know what that means. But some artery. Afferent arterial, glomerular capillaries, efferent arterial. So these capillaries are these, meshed structures of capillary. They're kind of beautiful in their own way, little walnuts. Here is a scanning electron micrograph of podocytes. So CB, cell body of a podocyte. P, process of the podocyte. And here, what do we call these dark lines between these fingers, finger-like projections from the process of a podocyte? Filtration slits. So the cartoon is actually a pretty good representation of what it looks like under a scanning electron micrograph. In cross-section, so this is a different type of electron micrograph. This is a transmission electron micrograph. So let's get oriented. CL, capillary lumen. CB, probably cell body of the podocyte. These here are the fenestrae in cross-section. And these here are the filtration slits of the podocyte in section. So you can see there are these microscopic holes between the capillary and what's going to be out here, the Bowman space. So this is how fluid flows. And not just fluid, but fluid and small molecules. This is microscopically small. A cell is probably this big on this scale, a red blood cell. So red blood cells and white blood cells are not filtered. They're too big to go through. Albumin. So you remember in cell-phys, we used bovine serum albumin as a standard protein. Let's unpack that word. Bovine comes from a cow. Serum comes from blood. Albumin. So do you have bovine serum albumin in your blood? No, you have human serum albumin. The point is you have protein in your blood, and that protein is too big to normally be filtered. So these filtration fenestrae and slits are so small and specially structured that they allow water, sodium ion, potassium ion, phosphate ion, chloride ion, glucose to pass through it, but not albumin, not a protein. There's a big, the molecular weight of glucose is, I don't know what, 60? Something like that. The molecular weight of albumin is 58,000 or something. It's a big difference.
So the red blood cells?
Cells cannot go through. Red blood cells, white blood cells, any cells in the blood. Those are the typical.
And also the protein?
Proteins in the blood cannot go through normally. If protein, so just to be, if you see one of the diagnostic tests of a well-functioning glomerulus is the absence of protein in the urine. If somebody does a urine test and they find significant amounts of albumin in the urine, it means there's something breaking down here in this filtration. It's filtering out things that are too big. It's a sign of glomerular disease. But it's not, and it depends. If you have gone through a strenuous exercise, there can be a period of time, because your blood pressure is high for a period of time, that you can get small amounts of albumin in the blood. So if you go to the doctor, and the doctor says, oh, you have some trace albumin, don't say, oh my God, I'm getting glomerular kidney disease. It could be just that you exercised pretty vigorously earlier that day. Okay, here's a picture from inside a glomerular capillary. What do we call these little holes that are on the inside of the glomerular capillary? Fenestrae. One fenestra, two fenestrae. Okay, so let's do a quick. Blood flows, this is an easy one. This one takes blood flows into the glomerulus. Okay. Blood flows out of the glomerulus. B. Urinary fluid flows out of the glomerulus. Urinary fluid flows out of the, well, it doesn't really flow, it flows out of the, this is not well designed. So I could see two different answers on this one, but most of you are answering it the way I would do it, that's D. But you could say, well, okay, forms the filtration barrier, forms the filtration barrier. Okay, so, like I said, this is a pretty easy one. Okay, I don't know if we have more, let's just see. Oh yeah, here's another one. Which of the following are freely filtered? So that's a jargon that I will use. Something is freely filtered. That is to say, easily filtered by a normal glomerulus. A lot of correct answers. All of them except for C. Glucose is small enough to be filtered. And we recover 100% of it. We want to keep our blood sugar. Why did we bother eating if we were going to piss the glucose or pee the glucose away? I mean, that would be wasteful. So we filter glucose because it's small, but we spend some energy recovering it. Okay, so now we're talking about the forces of filtration. I think this is the last slide. So, this is a cartoon of Bowman space. So what is this line representing? What is the thing that surrounds Bowman's space? The capsule. Bowman's capsule. I'm just trying to use the anatomy again, one more time. Bowman's capsule. What does this do? Two words. Second word begins with a T. First word begins with a P. Proximal tubule. Okay. What are the cells that separate the glomerular capillary from Bowman space? The cells that are represented in this white space? Podocytes. And then there's some endothelial cells as well. But the main thing that generates this filtration barrier are the podocytes. Okay, so what do you suppose P stands for? Pressure. What do you suppose GC stands for? Glomerular capillaries. So this is blood pressure in the glomerular capillaries. And blood pressure, so this, blood pressure pushes filtrate into Bowman space. Okay, so that one's easy. That one comes easy to everybody. Blood pressure pushes fluid from the glomerular capillary into Bowman space. But now we have to think about osmotic pressure, osmosis. How many of you have a sense that from like biochem or maybe economic, I don't know where you might have picked it up, an understanding of what is osmosis? Okay, so not very many of you. What flows? What is the thing in biological systems? What's the thing that flows under the influence of osmotic forces? Water. Water is what flows. And we're talking, water is one of the things that flows here. So, and what generates what causes osmosis? What causes water to flow from one place to another in response to osmosis? A concentration gradient. So if there is a high concentration of solute here and a lower concentration of solute here, then osmotic forces are going to push osmosis that way. I mean, it's going to push water that way. And so osmotic, so pi, osmotic pressure usually counteracts normal pressure. But we don't call it normal pressure, we call it hydrostatic pressure. So this is what you think of when you think of pressure. Hydrostatic pressure. This is osmotic pressure. Water tending to flow from a low concentration to a higher concentration. And why does this material here have a higher concentration than the material here? Albumin is the simplest direct answer. Remember, albumin did not get filtered. So as water is forced out by hydrostatic pressure, the concentration of solute, including albumin, increases. And that albumin, it's a solute, it's a protein, it's dissolved in water. It gives this, it's a solute. And so now there's the concentration of solute here is higher than the concentration of solute here because albumin stayed here and didn't go here. So now we have osmotic forces counteracting hydrostatic forces. So if we want to talk about the net pressure, it's the hydrostatic pressure in the glomerular capillary minus the hydrostatic pressure in the Bowman space. There's a little bit, that's this arrow. This arrow represents the pressure that builds up in Bowman space. It's not zero, it's got some pressure. It's almost always less than the pressure of blood pressure in the glomerular capillaries. But to do it right, we have to take the pressure, the hydrostatic pressure in the glomerular capillaries, subtract from it the pressure, the counteracting hydrostatic pressure from the Bowman space, and then subtract from it the osmotic pressure generated by albumin in the glomerular capillaries. This is the term that generates glomerular filtration pressure, which is the force that generates filtration. Net filtration pressure is blood pressure minus Bowman space pressure minus osmotic pressure. So that's the physiology and anatomy of some of the physiology, much of the anatomy that you need for the quiz. On Thursday, we will talk about a whole other concept called clearance, which will also be on the quiz. Okay, have a good afternoon, everybody. See you on Thursday.